Hello, and thank you for your interest in SAGA Diagnostics. My name is Lao Saal, and I am the co-founder and CEO of SAGA. We are a cancer genomics company based in Sweden and are focused on precision oncology through improved tumor tissue genomics and personalized cancer monitoring using simple blood samples. This is a really exciting area which we believe will transform cancer patient management and improve quality of life and outcomes. In this presentation, I will cover the ultra-sensitive detection and monitoring of circulating tumor DNA using our SAGA Safe and SAGA Sign technologies. As tumors grow, they release parts of their DNA into the tissues and into the blood and other body fluids. This so-called circulating tumor DNA is a new type of biomarker which contains all of the DNA mutations and chromosomal rearrangements present in the patient's tumor. SAGA has three technologies to quantify this ctDNA, called SAGA Safe, SAGA Sign, and SAGA Seek. These all have ultra-high performance, are proprietary and patented, have excellent validation data, and can be applied to any cancer type. SAGA offers our customers service testing, where your samples are tested in our lab, or we sell kits, which can be run in your lab. SAGA focuses on the cancer patient and how to stage patients and best predict their prognosis and identify drug targets, to monitor how the tumor responds to treatment, to detect residual disease and identify relapse at the earliest moment, and to make the promise of personalized oncology a reality. With our three platform technologies, we cover most of oncology. SAGA Safe is our ultra-sensitive method based on a patented improvement of digital PCR. It is multiplexable and ideal for companion diagnostics and monitoring a set of specific mutations. We service our customers, mostly biopharma companies and hospital and clinic labs, from our service lab, as well as providing kits to run in their lab. Our first CE marked kit was launched in October of 2020 for the EGFR resistance mutation T790M. Today, we have over 250 assays developed and a long pipeline of new CE IVD kits planned for release later this year and in the coming years. SAGA Sign is our second platform technology, which is a hybrid method using both NGS and digital PCR. In the first step, we do whole genome sequencing of the tumor to identify a so-called fingerprint of the patient's tumor-specific chromosomal rearrangements. And then in the second step, we use a digital PCR fingerprint assay to measure tumor burden in the liquid biopsy samples. This allows for personalized follow-up of therapy response, residual disease, and early detection of relapse. Customers are biopharma companies that use SAGA Sign to monitor patients in clinical trials in our Swedish service lab, and later we will also offer this in a CLIA CAP facility. SAGA Seek is our third platform offering, which is based on NGS and uses our customized library prep and machine learning-based bioinformatics to increase sensitivity and specificity. In addition to a pan-cancer gene panel, we will also offer customizable panels and tumor type specific panels, as well as a TMB panel for immuno-oncology. SAGA Seek will be launched later in 2021. What SAGA is well known for is our ultrasensitivity. We are able to push the limit of detection of our methods down to the 0.001% mu to allele frequency range. This is about 100-fold improved compared to other methods, which typically cannot go lower than 0.1%. With SAGA, you can unlock the potential in your samples, a potential which is hidden when you use methods with inferior performance. In this table, you can see the number of expected genome copies and approximate theoretical achievable LOD if only 2 copies of a locus are mutated. With other methods, 10 nanograms of input DNA is all you need because those other methods cannot detect anything lower than 0.1%. but a typical blood plasma sample contains from 10 to 50 nanograms of DNA per milliliter of plasma. In other words, from just one 10 milliliter tube, you might get from 50 to 250 nanograms of DNA, and it's quite easy to collect more than one tube these days. A number of factors can increase cell-free DNA quantities, as well as the amount of background wild-type DNA. So, with a single tube of blood, or two, there are low MAF mutations that can be detected with a true ultrasensitive method, such as those offered by SAGA. The major cause of poor limited detection of most mutation detection methods is error caused by the DNA polymerase itself. I can illustrate this by first explaining how standard digital PCR works. In standard digital PCR, a reaction is partitioned into many thousands of reaction compartments, such as the droplets shown here. When the PCR is performed, the compartments with the target DNA of interest light up with fluorescence. The intensities in the compartments are read, and each compartment is scored as positive or negative based on the intensity reaching a threshold. Since the partitioning was random at the beginning, one can use the ratio of positive and negative compartments and a Poisson correction to calculate very precisely the number of copies of target DNA that was present in the original sample. The issue is that the DNA polymerase error can lead to false positive compartments by the mutation of interest being generated during PCR as an error. When the DNA polymerase makes an error, shown here on the left compartment, the erroneous DNA is copied and propagated, leading to a false positive fluorescent signal. This is in contrast to SAGA Safe, where our patented method reduces the consequence of polymerase error. This is accomplished by having two phases of target discrimination, where a linear first phase means that any DNA polymerase error is not propagated into many copies, but stays as a tiny minority of the molecules in the compartment. So, when the fluorescent signal is generated in the second phase of the SAGA-safe reaction, the true state of the compartment, whether it is true negative or true positive, is maintained. The issue of false positives is illustrated clearly in an example like this. Here we have run pure wild-type normal DNA with a Bio-Rad KRAS assay on top and a SAGA-safe KRAS assay on the bottom. It's the exact same DNA and reaction setup, but as you can see, there are high-intensity false positive signals with Bio-Rad, but on the bottom, the SAGA-safe data is super clean. Here's another example for a dilution series of a PIK3CA H1047R mutation from 10% MAF down to 0% MAF, or pure wild type. On top, you can see the Bio-Rad assay has false positives, meaning that quantities below 0.1% are not distinguishable from wild type, and so the LOD is about 0.1%. On the bottom, SAGA-safe is highly linear and without the false positive issues can achieve ultra-sensitive LODs down to the 0.001% range. Due to lack of time, I cannot go into a full explanation, but SAGA-safe also allows for a more accurate quantification because SAGA-safe can distinguish the denaturation state of the input DNA. With standard digital PCR, one might get a quantification that is off by a factor of 2 if the DNA is fully double-stranded versus being fully denatured into single strands. Because SAGA-safe can quantify compartments and distinguish the number of strands within each compartment, the quantification can give you an estimate of the denaturation state and a corrected copy number. This is an important consideration which is overlooked by other methods. Importantly, SAGA-safe has been evaluated and is compatible with the 4 instruments tested thus far, from Bio-Rad, QIAGEN, Stila, and Comirnaty. So what does ultrasensitivity mean? Simply put, it means more mutations are detected. Here's an example of over 200 DNA samples from breast tumors, analyzed by targeted NGS to about 600x coverage, plotted in the bars, versus SAGA Safe plotted as the small diamonds. About 30% of cases were PIK3CA mutant by NGS, whereas SAGA Safe found those same 30%, but then an additional 50 or so more cases, down to a math of 0.003%. That is an increase of about 80% more PIK3CA mutants identified, which had the expected clinico-pathological correlates. This may be clinically relevant as Novartis now has a PI3 kinase inhibitor, Piqray, that is approved for metastatic breast cancer. We have other examples, such as here in breast cancers treated with neoadjuvant chemotherapy. We're still finishing the study, but we see that post-operative residual ctDNA is a very clear indicator of poor prognosis. And in melanoma, BRAF and NRAS mutations identified in the analysis of plasma from 77 patients is quite prognostic, with a hazard ratio of 7.8. Here we published a paper in acute myeloid leukemia tracking MRD using SAGA Safe, where we could identify residual disease months and years prior to clinical relapse, and at many time points where standard flow cytometric MRD was negative, shown in this example at the time points indicated by the red arrows. We launched our first CE-IVD marked kit in 2020 for the EGFR T790M mutation, which is a known resistance mechanism for TKI treatments in non-small cell lung cancer. This assay has a limit of detection of 0.003% and limit of blank of 0.001%. And in a study with Maria Planck at Skåne University Hospital, we identified T790M mutations in clinical plasma samples at very low mass, all the way down to 0.003%. And we had a perfect sensitivity and specificity when testing 70 blinded samples. And we have a pipeline of more assays coming. For example, a multiplex PIK3CA assay that covers the 40 most common mutations. This is about 3-fold more mutations than the comparable QIAGEN TheraScreen qPCR test. And our test has LODs about 20 to 100-fold improved compared to QIAGEN's test. Here you can see some example data, as well as the list of all 40 mutations that our multiplex assay covers. We also have EGFR assays covering all the common activating and resistance mutations, including one for pan-exon 19 deletion detection. Multiplex assay for the KIT gene, covering the 9 variants at the D816 position. A multiplex NPM1 assay covering mutation types A, B, and D with ultrasensitivity. And even a multiplex test that can detect about 100 different exon 14 skipping mutations in the MET oncogene. And as we work with digital PCR instruments with more laser channels, the degree of multiplexing will increase for all of our SAGA Safe assays. We are also working with copy number and have a HER2 amplification test that utilizes SNPs to increase diagnostic accuracy. And a genotyping assay for 6 common SNPs in the DYPD gene, which can be used for identifying patients that may be hypersensitive to 5-FU chemotherapy. With over 250 assays to date, we can develop custom assays for customers typically within about a month. So if you have an interest in collaborating on an ultrasensitive kit, please do get in touch. Changing gears here, we are also offering our SAGA-Sign method for ultrasensitive detection of minimal residual disease. The method involves whole genome sequencing to identify genomic rearrangements, followed by automated construction of a fingerprint assay, which can be then used repeatedly to test follow-up blood plasma samples. We've seen that chromosomal rearrangements are extremely stable during tumor progression, with about 90% shared in primary to metastasis in breast cancer, which is consistent with the notion that truncal rearrangements are formed early during tumorigenesis during the so-called telomere crisis. So every tumor has its own pattern of rearrangements, which we can follow and quantify in the blood plasma over time, and which we've shown has a very high sensitivity and specificity to detect occult metastasis in breast cancer, on average about 11 months prior to clinical relapse, and in some cases up to 3 years earlier. The ctDNA quantity measured by SAGA Sign is also highly predictive of recurrence-free survival and overall survival, and also significant in multivariable analysis. We offer this type of testing from our service lab and are planning to launch our own prospective studies in the near future. Finally, we have our SAGA Seq platform, which, as I mentioned, we will offer various kinds of gene panels, from large pan-cancer panels to smaller cancer-type specific panels, a TMB panel, as well as flexible plug-and-play content panels. With our proprietary improvements, we will achieve performance down to the 0.01% mass range, and this will be available later in 2021. In summary, SAGA offers specialized tumor genotyping and liquid biopsy analyses that are useful across a cancer patient's journey. We help biopharma, KOLs, and scientists to perform research, clinical trial support, and assay co-development. We can offer customized solutions and kits and work with all types of samples. And we are seeking new collaborators, partners, and early adopters, and even clinical laboratories who, like us, see the promise of improved diagnostics to revolutionize how cancer patients are treated and monitored. Thank you very much for tuning in and for your interest in SAGA Diagnostics. If you'd like to find out more about how SAGA can help you in your research, in your clinical trials, and drug development programs, please visit our website at sagadiagnostics.com or send an email to info@sagadiagnostics.com. Thank you very much.